Today, we are going to explore one of the most fascinating and delicate processes that occur inside the human female body, implantation, which is a crucial step in early pregnancy. Let's begin from the moment of fertilization and follow the journey step by step. Fertilization takes place when a sperm cell fuses with the secondary oocyte or ovum in the ampulla of the fallopian tube. This ovum is definitive, meaning it has a haploid number of chromosomes. Once fertilization occurs, a zygote is formed. This is a unicellular embryo that carries a diploid set of chromosomes, means 46 chromosomes. Around 30 hours after fertilization, the zygote undergoes its first division through a process called cleavage, forming two cells known as blastomere. Cleavage is a series of rapid cell division that occur without growth. It proceeds as follows: first 2 cells, then 4 cells, followed by 8 cells. Finally, it reaches the 16 to 38 cell stage called the morula. Around the third day, the morula then enters the uterus, where it develops into a blastula, a hollow ball of cell. The question that arises here: what is the difference between cleavage and mitosis? Let's clarify an important concept. In mitosis, cell division is equal, meaning the daughter cells are of the small size. In contrast, cleavage involves unequal cell division, producing smaller cell. In mitosis, the overall size of the cells and the embryo may increase, but in cleavage, the total size remains the same because of the zona pellucida, a protective layer that prevents growth during early division. By the way, let's learn about the blastula stage. When the blastula forms, the zona pellucida breaks down, allowing the embryo to grow in size. The blastula contains a fluid-filled cavity called the blastocele. It has two distinct cell groups. One is the outer cell layer called the trophoblast, and other is the inner group of cells called the inner cell mass, ICM, or embryoblast. Around the 7th day, the blastula attaches to the endometrial lining of the uterus. This marks the beginning of the implantation. After the blastula attaches to the uterine wall, the trophoblast differentiate into two layers: syncytiotrophoblast, the outer layer, which is multinucleated and helps in embedding the embryo into the uterine wall. It also secretes beta HCG, human chorionic gonadotropin, the hormone used in pregnancy test, which become detectable by day 13. Cytotrophoblast, the inner layer, which give rise to extra embryonic membranes such as the amnion and chorion. Amnion, which became the amniotic sac filled with amniotic fluid that cushions and protects the embryo. On the other hand, chorion, which plays a role in forming the placenta. By the way, let's look at the inner cell mass and gastrulation. The inner cell mass or embryoblast continues to grow and undergo a process called the gastrulation. During gastrulation, the cell differentiate into 3 germ layers, which give rise to all tissue and organs of the body. One is ectoderm, forms skin, brain, nerves, etc. Number 2 is mesoderm, that forms muscle, bone, blood. And number 3 is endoderm, that forms internal organs like the gut and lungs. Now, let's understand the structure and function of the placenta. The placenta is a temporary organ that develops in the uterus during pregnancy. It attaches to the uterine wall, usually at the top or side, and connect to the baby through the umbilical cord. After the baby is born, the placenta is usually expelled from the mother's body within 5 to 30 minutes. If the birth is by caesarean section, the placenta is manually removed by doctor. Now let's explore the component of the placenta. The placenta has two components. One is maternal part, other is fetal part. Maternal part derived from the uterine tissue, and fetal part derived from the trophoblast of the blastocyst. Keep in mind that in human, the placenta is of the chorioallantoic placenta, meaning it forms from two embryonic membrane, the chorion and the allantois. It is typically about 9 inches in length. Let's discuss the function of the placenta. Placenta, it allows for the exchange of blood between the mother and fetus without mixing the blood itself. It transfers oxygen, glucose, and essential nutrients from the mother to the baby through the umbilical cord. It acts as a filter, removing carbon dioxide and waste product from the baby's blood. It produces hormones necessary for the pregnancy, including human placental lactogen, estrogen, and progesterone. It helps transfer antibodies from mother to baby, giving the newborn passive immunity just after birth. In summary, implantation is not just a single event but a complex, well orchestrated biological process. From the union of sperm and ovum to the formation of the placenta, every step is essential for the nurturing and protection of new life.